Diffusion is defined as the process by which molecules intermingle as a result of their kinetic energy of random motion. The rate of diffusion can be affected by the size of the molecule or the temperature.
Passive transport is a type of diffusion that doesn't require energy. It allows small hydrophobic molecules to diffuse through a phospholipid bilayer. Active transport is another type of diffusion that moves molecules against a concentration gradient across the plasma membrane, transporting them in or out of a cell. This type of diffusion does require energy.
Osmosis is defined as the net movement of water molecules through a semi-permeable membrane from a high concentration to a low concentration. Osmotic pressure is the pressure in a concentrated solution that keeps water from moving into the membrane.
Diffusion is a generalized term associated with the movement of particles. Meanwhile, osmosis, a type of diffusion, is only associated with the movement of water through a selectively permeable membrane. There are many ways we can associate diffusion and osmosis with the human body. One of the most important examples would be when it comes to how IV solutions are created. The purpose of IV solutions is to have the ability to be directly injected into a human harmlessly, which means that it cannot be 100% water, or a person's blood cells would burst upon injection.
To represent osmosis and diffusion in real life, we conducted 3 experiments. For this experiment, we used 2 different solutions. The first solution shows 10 drops of red and blue ink being added to a glass of water. When the ink touches the water, it diffuses throughout the glass. Next, we added 10 drops of the same colored ink into a glass of vinegar. When the ink reaches the vinegar, it diffused throughout the solution at first, but then began to seep towards the bottom of the glass, where it remained in a clump.
For this experiment, we placed red and yellow gummy bears into 3 different solutions. From top to bottom, the solutions are sugar water, tap water, and salt water. We monitored these for 8 hours. The experiment showed in each cup that the dye from the gummy bears diffused out into each solution. The rate of diffusion increased from sugar water to salt water.
For this experiment, we put 4 eggs into vinegar to strip the shell and reveal the inner membrane. After the shell was dissolved, we weighed the eggs and found their starting weights to be 95.21 grams, 103.52 grams, 94.82 grams, and 98.43 grams. Next, we put the first 2 eggs into water and the second 2 eggs into corn syrup.
The egg membrane soaked in these for 24 hours. After being soaked in the water, the weight of the eggs increased from 95.21 grams to 98.37 grams, and from 103.52 grams to 106.26 grams. In the corn syrup solution, the egg weight decreased from 94.82 grams to 61.74 grams, and from 98.43 grams to 57.61 grams.
This experiment shows how osmosis is happening when the water is diffused through the semi-permeable membrane of the egg from a high concentration to a low concentration. This is why the eggs in the corn syrup solution shrank while the ones in the water grew.